Welcome to the Axial Podcast. Axial is an early-stage investment firm based in San Francisco. We partner with great founders and inventors investing in early-stage life science companies often when they are no more than an idea. Axial is fanatical about helping the rare inventor who is compelled to build their own enduring business.
Okay. Hey, Peter. We're recording now. Great chatting up for the last 10 minutes. Thanks for doing this. Awesome to have you on the podcast. And we'll do this 2 years from now and just get the update. But maybe you could start with a quick introduction about you and your work, and then we go from there.
Sure. Yeah. So I'm Peter Turnbaugh. I'm a professor in the Department of Microbiology and Immunology at the University of California in San Francisco.
Cool. And so maybe we can transition towards first of all, how you got into science before we talk about your research and your lab and whatnot.
Oh man, yeah, way back.
I'm gonna learn a lot here. I'm really familiar with your work. And thanks for teaching me some papers.
Yeah.
But I'm gonna learn a lot in this conversation myself.
Yeah, I was never really a born scientist. I think I kind of, I went to liberal arts school for undergrad called Whitman College, which is in a small town famous for its onions in Eastern Washington. And I didn't really know what I wanted to do with my life. I was a typical kind of liberal arts student, just kind of exploring. And I kind of just got interested in science classes without really thinking about whether or not it was a career. I was, just curious about the topics. And I also, I was kind of turned off by the more humanities classes where there was a lot of blue book exams. And so, I didn't really like, I kind of felt like the grades were arbitrary. I really liked the fact that in science, it seemed like the grades meant something. And so that just kind of steered me towards majoring in science. And I wasn't even really planning on doing it as a career until I was a junior. I did a study abroad in Australia, and I got a chance to do some research in the stem cell lab there, led by Peter Turnbaugh, and that just kind of ignited my excitement about basic research, and I had a really fun time, and I ended up staying for a whole year and just doing research the whole time.
So in Australia,
I just really liked it.
What was the spark? Some scientists just find out they're really good at it. Some people find, I find a common thing is a lot of scientists get a kick out of knowing something no one else knows. But what was the trigger, in that stem cell lab that kind of got you hooked?
Yeah, it was exactly that. I think in the coursework, it all seemed like just a series of facts that we were trying to memorize. And, I was really good at memorizing things. So I like that. But yeah, going into more of a research lab, the lab at the time was trying to, take embryonic stem cells and make them into neurons that would produce dopamine and be useful for treating Parkinson's. And so I was, kind of immediately excited by kind of, the potential implications for changing how we treat disease, but also just the fact that nobody can do that. And so it was really exciting to be trying to do something that nobody else had done before. I never really thought of that in terms of my basic, coursework.
Cool. And so maybe we can then transition towards your graduate career. And so you have either one of the luckiest careers as a graduate student, or one of the most thoughtful and mastermind graduate careers ever. So let's figure out what it was, maybe somewhere in between. But how did you make that transition from, college, doing exploratory research, getting experience in laboratories, to then making a decision to go to grad school? Not, or did you do something in between? And how did you figure out to go to St. Louis?
Yeah, I was after that year abroad, I was really excited about doing research. So, I applied to grad schools, but, it's even more competitive now. But, I was, I had 1 year of research experience, and a lot of people are coming out with 4 years of experience and, lots of publications, and maybe even extra years after undergrad. Yeah. And so, I applied to a lot of schools and got rejected from almost all of them. Including UCSF. And so I basically applied, on the whole Western, all the way from Washington down to Southern California, and just got rejected from every single school. And so, the two I got in, I got into two schools in the Midwest, and I ended up at WashU in St. Louis. And I'm really happy that happened. Although, at the time it seemed like, I was moving into the middle of nowhere or something.
I've been to St. Louis, and it's a beautiful campus, but the city's pretty rough. And so it's just like, I remember somebody told me in St. Louis, don't go past the street. There's like a street you don't go past.
Yeah, there's a lot of borders you have to know, and
yeah, exactly.
Actually, my first weekend there, I had driven my car. I had this beat up Dodge Neon, and I parked it next to the place I was renting, and then I flew back to my parents' place to get some like textbooks and stuff.
Yeah.
And over the weekend, I got a call on my phone, and the guy said, hey, did you park in my yard? Like, no, I parked in the street. And he's like, well, your car is in my backyard. And so, after, a couple of days in the city, my car had been stolen and dumped in somebody's yard.
Man.
So yeah, we definitely, it was a dangerous place, but the school was amazing, and I really am lucky that I ended up there.
And St. Louis is awesome. Also, just to give a positive on St. Louis, you can be a grad student there and also buy a house. I know people like that. Like they live in some of these smaller towns and go to grad school, and they just have a mortgage. It's like insane for like people who go to UCSF or Stanford for grad school. So
yeah, the now wife bought a house just right south of campus, and so we lived. We could walk to the lab, and we had lots of space, and
plus or minuses.
It was awesome.
You're at St. Louis, and this is like 2000, I think 6 or 7, or
I got there in 2004.
2004, okay.
Yeah.
And so you're there in 2004. St. Louis had become like the epicenter of sequencing, mainly because no one else wanted to do it, from my understanding. They did the grunt work for the Human Genome Project and a bunch of other stuff. What was your experience in that backdrop, and how did you pick labs, and how did you figure out to ultimately work with Jeff Borden?
Yeah, that was kind of, so I was really, really excited about genomics, and, also being at WashU, with all the sequencing happening. I was really interested in computers, and so, I wanted to do some kind of bioinformatics.
Cool.
And so I ended up kind of rotating, we had, in the program I was in, we had to rotate in 4 different labs. And so I did started in kind of yeast transcriptional regulation, and then I went to a zebrafish lab, and then ultimately for the 3rd rotation went to the Gordon lab. Cool. And so I wasn't even really thinking about microbes at the time. I just wanted to look at genes, basically. but Jeff, really had a lot of was very like forward thinking, and that at the time everybody was really focused on eukaryotic genomics, and he really thought that DNA sequencing would teach us a lot about the microbes that live in and on the body, and he thought that would be really important for medicine. But most people disagreed, and thought we should be spending our money sequencing more eukaryotic genomes, and so he was really like pushing towards studying microbes at the time.
So maybe that's a really important part of history. Even I'm interested in, was metagenomics a term? Was it a thing that was like popular, or was it kind of criticized? And like it seems like around 2010 metagenomics became more mainstream. But at least at the time you're in grad school, what was kind of the status of the field of like sequencing microbes with like just shotgun tools, or just kind of different types of like more agnostic measurements?
Yeah, I mean, I would say even at the time, like sequencing isolate genomes of microbes was rare. So like this happened right before I joined the Gordon lab. But Abigail Salyers, a really famous microbiologist, had developed this bacterium, Bacteroides thetae omicron, as kind of the workhorse member of the Bacteroidetes phylum that is in all of our guts. And and they'd done all these really nice kind of basic science studies and learned about how it eats sugars and does all sorts of interesting things, but nobody had sequenced the B theta genome. And so a few years before I joined the lab, Jeff was sort of begging the Genome Center to sequence B theta, and they kept turning him down. And so ultimately he had to, I think he got like a leftover sequencer that was gonna be thrown out, and they sequenced it in the lab themselves, and then that kind of became the first reference genome for one of the major players within the gut microbiota.
Wow. Okay, I didn't know that.
So yeah, I mean, I think even isolate sequencing was really rare, and then when I started in grad school, really nobody was doing metagenomics. It was, I mean, there were people in the kind of environmental microbiology community, applying that kind of approach to, study the ocean and other aquatic systems, and studying soil and acid mines and all these interesting samples, but not much going on in terms of the human body.
If my understanding, a lot of it was like microbial ecology, and then like you said, people were kind of trying to study microbes in the context of like ocean metabolism or stuff like that. And so, okay, you're in Jeff Gordon's lab.
Yeah,
Gordon's lab.
Yeah, and at that time, like one of the big projects was the Craig Venter had bought a sailboat, and he was sailing around sequencing seawater. So I mean, there was a lot of really exciting environmental microbiology projects. But
I think the Craig Venter thing was getting a little. It was very interesting, but he also, from my understanding, it was on a yacht, and he had some really like. It was. I don't think it did the field too much good in terms of like perception, and so but maybe you can. I remember also the class you're part of, Peter Gurgis taught. He did really cool research on like biogeochemical cycles,
right? And looking at the deep sea, and
it was some really cool work. And so
basically all of these microbial ecologists were going to really cool places, and
exactly.
They weren't studying the gut.
I remember I think Michael Gilmore, somebody told everyone, if you want to be a scientist, try to like be Darwin or get on a boat somewhere and do PCR in the ocean. Don't do PCR in a lab. And
right.
That's a pretty good argument. But maybe we can. Okay, so how did you end up wanting to work with Jeff at all the labs you rotated in? And for that initial project, did you just jump all in on like sequencing microbes, or was it pitched to you, and did you have to like work your way towards it?
I mean, I think the thing that really attracted me to Jeff was really his attitude. He's just, I don't know if you've ever met him, but he's an incredibly kind of charismatic and engaging person. He's you just, as soon as you meet him, you want to spend more time with him. And he, even when I was rotating in one of the other labs on the floor, he would always walk by my bench and say hi to me, and he called me professor for some reason. I just he's like somebody you want to be around, and he's also he has such a good vision for the future that it's really inspiring. And then the other thing that really attracted me to the lab was that Ruth Lay had joined as a postdoc, and she came from Norm Pace's lab, and brought with her all of these tools for measuring microbial communities, and also a lot of knowledge about ecology and kind of how microbes communicate with each other and the environment. And so it's definitely kind of a right place at the right time kind of situation.
Cool. For context, Norman Pace and along with Carl Woese are kind of like the key figureheads in microbiology, and so they have this really cool. I remember I read this review, really fancy, something that the Pythias, and had some really fancy Greek name. I'll link it in the description, but it's okay.
Lucky for us, Norm had been doing this like amplicon sequencing stuff since the 70s.
Exactly.
But he really had not looked at humans. So, he kind of left a big hole in the field for.
Maybe we can give context in terms of like, the real revolution happened in the 70s where people were sequencing ribosomal DNA of microbes who didn't classify them, and that led to like a whole reclassification of like microbial life. and the discovery of archaea. And so maybe in the context of using these tools and those methods for the human microbiome, what was kind of Jeff, yours, and Ruth's like vision and plan to probe the human microbiome, and like what kind of like hypothesis did you have in the beginning?
Actually, the first project I worked on when I was a rotation student was, I guess, bigger than that. Like Ruth really wanted to get a sense for what the gut microbiome looked like in many different mammals. And when I started, the first day in the lab for me was, Ruth had already gone to the St. Louis Zoo and collected poop from a lot of different animals. And so she had a -80 that was just full of animal poop. And she said, "Here you go." I mean, she gave me a protocol for how to extract DNA, and then do 16S amplicon sequencing, and just set me loose. And then part of the time, I also we did a second expedition to the zoo, so that was really fun. We got to sample more animals, and then later on extended that study to the San Diego Zoo and to Africa. And so it was a really fun kind of like real microbial ecology kind of survey project. And then that kind of got, helped me learn the basic techniques and start thinking about microbial communities. And then for my own kind of thesis work, like, even I think when I was, or shortly after I joined the lab, like, Ruth and Jeff had been really interested in a long time in kind of how microbes affect energy balance.
Yeah.
And so they'd been studying this, I guess a really key paper that had happened, or I guess came out like around the year I joined the lab, was led by Frederick Beckett. And he did a really simple experiment where you just compare germ-free mice to mice, so mice that are living without a microbiome, to mice that have a microbiome, and he found that it really affects how fat the mice are.
Yeah.
And so that showed that the microbiome somehow plays a role in controlling body fat. And so Jeff immediately thought, oh, maybe that means that microbes can play a role in obesity, and we sort of turned to modeling that in mice, and also we're able to look at that in humans.
Cool. And then so you published you and Ruth published some key papers.
Mm-hmm.
2006, 2009, kind of establishing a core microbiome for obesity. And could you describe like that work, but more importantly, the challenges and the pushback you get? Because when you measure a large ensemble of microbes, and how do you establish which species and which populations are actually driving the phenotype? So how did you? Especially this is like 15 years ago.
Mm-hmm.
What was seen easy now was really hard then. So how did you deal with those challenges of establishing the core microbiome and linking it to obesity?
Yeah, I mean, I think it was both exciting, but also, kind of wild to think about in hindsight that like we didn't really have any, we didn't even have any protocols for any of this stuff. So like, we didn't know how to actually generate the data. There were no like software tools to deal with the data. So, we were mainly doing like sequencing based approaches, either like sequencing just 16S or sequencing the whole microbial community. Nobody had kind of figured out how to do that well. And so, we were kind of just like doing everything from scratch. And, I'm sure making a lot of mistakes, now, and, a decade later, there's, all sorts of fancy bioinformatics tools that you can use. But, that was really fun because, there wasn't really, you didn't have to follow a checklist. It was just like everything we did, we could do whatever we thought seemed right, and then I guess like conceptually, we kind of touched on this earlier, but we got tons of pushback just with the idea of studying microbial communities. And I kind of still face that every day.
Really?
You know,
Jeez.
I think that the world obviously is excited about the microbiome, but I think that a lot of scientists are really worried about the complexities once you have like many different microbes interacting and they also have a host. And so there's this perception that you're never going to be able to make sense of anything. And so that, that's what we faced from the very beginning. People said, you're wasting your time doing metagenomics. You should just be doing, genome sequencing, just focus on a single organism.
Yeah.
There's always this push towards like reducing you down to something that's really tractable, and then we also got, a lot of pushback in terms of like the specific things we found. So like one of the things I found as a graduate student was that, eating a high fat diet really rapidly changes the microbial community in the gut. And some of those changes can then, increase body fat in the host. And, I remember the first time I presented that at a graduate retreat. The first question was like, "Oh, that's obvious. Like, obviously diet matters." And so, like, there was sort of both like the idea that it was going to be too complicated, but also like that the things we were studying just weren't, novel or interesting. And so yeah, we had to kind of just. I mean, I think what I had in my corner was Jeff, who was always like so encouraging and like, he, whether or not he believed it or not, would, no matter what we would do, it would always be exciting, and he was just a really good cheerleader, and so that always kept us going through all the negative feedback.
Awesome. So when you're like in an uncharted scientific territory, where there's no protocols, there's no playbook, who do you lean on? Do you lean on like some sort of text or prior literature, and just kind of get inspired by? Do you lean on certain mentors, or maybe do like a peer like Ruth? But what was kind of the like, over that course of those few years early on, like how did you manage to navigate through this maze of like unknown territories, which is like extremely hard in science, because most science has like a protocol. There's already kind of some body of literature behind it. You're kind of pioneering a new field. How did you do that from like what resources did you have to rely on to like just get a sanity check, to get verifications, to get people to, like you said, be encouraging to move forward?
Yeah, I think like I don't know, maybe it's a strength of mine, but I've never been shy about like asking people for help. So like, I had Ruth that I could talk to every day, and then I bothered a lot of other faculty all the time. So, we were in the genome center, and so, there were people like Sean Eddy that were just nearby that, knows a lot of computational biology, and, people that I could just pester, for advice. And then the other thing that I did a lot of was like reaching out to people in the microbial ecology field, the environmental microbial ecology. And so, one of the guys who I still talk to is Ed DeLong, and he was really a pioneer in, studying microbial communities in the ocean, and really graciously respond. My wife, or girlfriend at the time, we were going on a trip to the Northeast, just a road trip.
Yeah.
And I was going to Boston for the first time, and so I emailed Ed, he was at MIT at the time, and said, "Hey, I'm going to be in town. Like, can I pick your brain about, how to do metagenomics?" And he replied and like showed me the lab, and like, and then has continued to support me and write letters of support for decades. So just to like kind of relying on these other people that I thought had kind of done similar approaches, but in other systems.
Did you when you reach out to people get help? Were people generally encouraging, like keep on going, or did you get like pushback, like, hey, this is kind of stupid, this is a stupid idea, I shouldn't do it? Because often like science, even from their mentors, get like this feedback that don't do that project, it's never going to work, and then they do it anyways and it works. That's like a common motif in science. Did you get a lot of that, or did you get a lot of like, oh, it's a great idea, keep on going, Peter? Yeah.
It's definitely a mix. I mean, I think the environmental microbial ecologists were like on board, they were super excited,
Cool.
because they're like, they know that the communities matter, and they were already drinking the Kool-Aid, I guess.
Cool.
But a lot of the other people I would talk to that were, maybe like on the human side, or, hadn't really thought about microbes before, were very discouraging. Some of them I kind of kept trying to get them engaged, but I mean, you kind of get in the cycle of like trying to do whatever you can to make them, I mean, I think it was motivating in a sense that like, I wanted to do better. Yeah, I still am trying to do better to make the people that are skeptical, think that this is legitimate.
Makes total sense. Yeah, I didn't know Sean Eddy was at St. Louis. Like, he's like maybe the best or one of the best bioinformatic tool developers ever. And so, like, you're really at the right place at the right time. So
yeah, definitely.
like,
Yeah.
definitely.
Well, and I think another nice thing about WashU, which is also true at UCSF, is like, people are really willing to talk to you. I mean, even if they're, even if they're negative, they're willing to like sit down with a grad student and talk to them. So yeah, I had a lot of free access to lots of really good faculty.
And so around grad school, you're trying to grad school. I don't know when the microbiome got really popular in popular culture. I don't know, 2009, 2010. I don't know which year. You published this work, you do your thesis. What was the what was the key moment in your graduate career that made you like realize, hey, maybe I should like do this myself. Maybe I should start my own group versus a bunch of other options you probably had.
Yeah, I mean, I was always kind of curious about industry, so like I was part of this bioentrepreneurship club when I was in grad school. But I think like the research I was doing was going well enough that like I never really thought about that as a serious alternative. Like, I mean, it was really from like that first year in grad school, I was all in. I just like, I mean, I wanted to like keep going as far as I could go. And so, if they would have me, I wanted to be faculty. So yeah, I never really like considered alternative careers.
Yeah, when you definitely have like when you pioneer in a scientific field, I think you should be a professor.
Yeah,
be honest.
Yeah,
and then somewhere in the middle. There's always but like if you're productively discovering, publishing papers and like something that's unknown, yeah, definitely, it's easy. So how did you pick Harvard? So after grad school,
I mean, I think my biggest fear was that the whole field would disappear.
Really? At the time, you were scared that like this would just kind of get old, because like I would think you're around the time you're in grad school, the human microbiome, just microbiome in general, is getting a lot of attention.
It was, yeah. And there's the big NIH microbiome project that was launched, and so there was a lot of excitement. I mean, I think like we've all, if you think about the history of microbiome research, there was a huge, not as big as today, but there was a big kind of burst of activity from like in the 60s and 70s, 1960s. And then the field really disappeared. And so, I was kind of deathly afraid that would happen again. And I still don't really know why it happened last time, but so far it seems like we're pretty stable. But
it makes sense. I don't know, some of these older microbiologists, like Carl Woese in particular, were kind of ornery. Like they just weren't willing to like engage the public. I don't know.
Maybe, I don't know. Yeah. So yeah, hopefully the ball has been rolling fast enough that nobody can stop it now. So.
Success. I think there's some success, at least. Like C. diff. That's on the product side, and scientifically, I think there's a lot of cool discoveries, and you're kind of leading that. So I think there's pretty good momentum where it's real. It's reproducible. That's
right. Yeah.
And so.
Well, and I think also this time, which is very different from the 70s, like we've now kind of infiltrated all sorts of established fields. So, I think like there's a little bit of microbiome in kind of every area of science.
Yeah.
So it's gonna be harder to get rid of us all. You know?
Okay, so you're at St. Louis, you get your PhD, wrap your thesis, and you become a fellow at Harvard, so running your own group.
Mm-hmm.
How did you make that choice? Why become a fellow versus get a postdoc versus try to become a professor? Why a fellow and at Harvard of all places?
Yeah, I mean, I think I was like very excited to have my own research group. And yeah, I had heard about these programs you can apply for where they'll give you a lab right after your PhD. I mean, I was definitely thinking of postdocs as another option. But I put in applications to Whitehead, the Bauer Fellows Program at Harvard, and the UCSF Fellows Program. And I actually ended up interviewing at Harvard and UCSF, and UCSF rejected me. So sort of, attempt number 2. But I'm very persistent, so I got there eventually. But yeah, so Harvard was basically it was either that or a postdoc. But I loved it. I mean, I was thrilled that I got in. I mean, it seemed like a miracle at the time. It was like, I got my own lab space and over $1 million to do research. 5 years of protected funding, and the best thing about the Bauer Fellows Program too is like there's really no other like obligations. You don't have to teach or like there's no committees. You don't do any of the other parts of being a faculty. You're just you just have money to do research, and they let you do whatever you want.
Yeah, totally agree. I think I'm a big fan of these fellowship programs. The coolest one is the Harvard Fellows or whatever, like that, because they have these really fancy dinners.
Well, that was the other program. So
yeah, so that's kind of the one to
the Bauer Fellows, we didn't get the fancy dinners.
Yeah, you don't get the fancy dinners.
The junior fellows, the Harvard Fellows get the dinners.
Exactly. But what was the kind of
program?
Exactly. So as a Bauer Fellow, how did you learn how to start a group? Did you like get Jeff to help you? Did you like take class? How did you like learn how to like recruit people, lead them, and set like a research vision? Not just versus being a grad student where you're often just doing the work and participating, but how did you learn how to like lead a group?
Yeah, I mean, I've always been a fan of kind of learning by doing. So most of it was just kind of on the job training, I guess. I mean, I did try to do some reading. Like I got the At the Helm book that a lot of people have read, and I forget who the author is. And then I downloaded the Making the Right Moves guide from HHMI, and I tried to prepare myself a little bit, but but yeah, most I was really flying blind, I guess, for the most part.
And so when you set your group up, what was the kind of the initial research approach? Was the niche research like focus? Was it intersection of microbiome and diet, or was it something more broad and narrow? But early on, what was that kind of like focus area?
Yeah, it's interesting. Kind of I was just recently looking back at my application, so I had to write like a 3-page research plan, and then the whole idea when I started in the fellows program was I wanted to switch from thinking about diet to focusing on the role of the microbiome in pharmacology. And so I was really interested in the sort of almost a century-long history of evidence that the microbiome affects drugs, and this was also kind of inspired by some of the early work we did sequencing the human microbiome. We found all these genes for xenobiotic metabolism, and xenobiotics are defined by substances that are not normally in the body, and so unlike something like carbohydrates or amino acids, like it just seemed really weird. Why would bacteria that live in the gut have evolved all these enzymes, and other proteins that deal with compounds that are not supposed to be in us. And so, I was really interested in kind of studying how microbes interact with drugs. And so in the beginning, when I started the lab, I was trying to set up kind of high throughput screens where we would look for kind of induction of gene expression or metabolism of drugs. and using kind of methods where we could clone DNA from different bacteria into a host like E. coli. And then we kind of, I guess, as we were trying to develop those methods, realized that there wasn't a lot of like positive controls we could use. Like there wasn't really enough like known about how any bacteria interacts with the drug, aside from antibiotics. And so that kind of forced us to narrow in and focus on this cardiac drug called digoxin, and how it interacts with one particular member of the gut microbiome.
Interesting. So, and how did it like? I didn't really actually. So you had a key paper in 2013 out of the group. It was more focused on how diet affects composition of human microbiomes. Maybe you could talk about some of the work that came out of your group as a fellow. So it seemed like some of it was focused on diet, and then the xenometabolism and stuff I'm not as familiar with.
Sure, yeah. We've never been able to kind of kick the diet side of the lab. So we're still working on diet today. We're mainly working on ketogenic, kind of high-fat, low-carbohydrate diets. And when I was a Bauer fellow, we did do additional work, kind of following up some of the stuff I'd done in grad school, looking at high-fat diets, as well as like other aspects of obesity. We had some work together with the Kaplan lab at MGH looking at gastric bypass surgery. But yeah, I think the paper you're referring to is part of what, so we had done a number of studies at the time, showing that in mice you can really toggle the microbiome using diet. So if you switch from a low to a high fat diet, within a day you get this massive difference in the gut microbiota. But nobody had really shown that same kind of switching in humans, and so we wanted to do a diet intervention study in humans to try to see if we could push the microbiome in different directions. And I was luckily kind of connected with General Mills, the food company, and they, at the time they were really excited about the microbiome, and so they provided some funding for us to kind of design and do our own human intervention study in the lab. So we actually that paper was all done by us. We designed the diets and we cooked them. And we recruited all the people, and then we fed it to all the people. So we were kind of our own nutrition study center.
What would you feed them? Like in a soup center, or like they would come to the lab, or you go to them?
Yeah, in our building. Yeah, so we some of the meals are in the cafeteria, and some were kind of up in the lab, like the common area.
Okay, I didn't read the methods section. How did you? I didn't read the methods.
Yeah, I don't think it says it.
That's a pretty good method. How did you ensure like compliance? You're like you have to monitor. You're doing. It's like you're running your own trial yourself. That's like a lot of work.
Well, yeah, it was a lot of work, and yeah, in hindsight, now we're collaborating with people that do that for a living. Yeah. So I don't know if I would do it again, but the nice thing is we were we were there for every meal, so everybody, it was only the diet intervention was only 5 days. Yeah. So like, people would come in and eat together. So, we could actually see them eating, which is really different from how most diet studies are done. You typically can't, usually they're at home eating, So yeah, I think it had some advantages in terms of that. And I think also it was both a strength and a weakness of the study, but the people we recruited, we kind of recruited from within the Harvard science community. So, they were pretty invested in the study, which you don't always get if you just have a general recruitment.
That seems to be a common theme, at least early on in human microbiome research, is that like the participants are the scientists. Or close, and then that's probably changed over the last decade. Any funny stories of like self-experimentation? Or I remember you kind of walked through that paper where the rumor is the lead authors were the samples over a year. But any like funny stories of like microbiome researchers, like stealing poop maybe, or was there a poop thief, or something crazy?
I don't know. Yeah, I don't know if anything particular comes to mind, but yeah, it was definitely interesting to like kind of see, that particular paper we're talking about. We had decided to really push the microbiome, to have a strong dietary intervention. And so one of the diets was all vegetables and the other one was all meat and cheese. And so we went to kind of the two extremes of diet. And so, it was kind of funny to see like, our participants kind of fell into camps, who's on the veggie side and the meat side. So, I love meat. So I learned that I'm happy to eat meat for a week, but I'm pretty miserable on veggies.
That's hilarious. Okay, so you had Harvard, and publishing research, running a lab, and then after your fellowship ends, you have to figure out what to do next. I'm sure you had a lot of offers. I think Harvard offered you. How did you end up choosing UCSF to kind of become a professor and get on that journey?
Yeah, I mean, I think I was, like I said, I'd been trying to come here ever since grad school, and so I had always, I guess, been enamored by San Francisco. Like, I really love the city. I grew up in Oregon, so I wanted to come back to the West Coast if they'd have me. And as like UCSF is just such a powerhouse for biomedical research. It's number one and the number one most funded public school.
Yeah.
It also, I think, has a really nice balance of like really amazing research, but also the kind of people are very modest and very willing to collaborate, and just the environment is very positive. And so I felt like it was somewhere that I would really fit in.
Cool. Yeah, UCSF is awesome. The one is like probably the best translational researchers in the world. Maybe Scripps is up there too, but like the one thing about UCSF is there's no college kids there. Like you're definitely, as a grad student at least, you're the dumbest person on campus. You know what I mean? Like at least when you go to like Berkeley or Harvard, Stanford, you have college students, and at least they're still the noobs. The grad students are now the low level, and they have to like just it just kind of creates a different type of dynamic where there's no college kids, so it's kind of a little more intense sometimes. I go in there, but then it's very collaborative too. So it's not
right.
Like,
I agree. It is a very different feel. And it doesn't really feel like you're at a college.
Yeah.
Really.
Yeah.
I mean, it's very like research focused, mission driven, and like, there's not a bunch of people playing frisbee or what you would get if you go to a normal. Yeah, exactly.
Very professional. You're here to get to work and make an impact, and not. It's not like exactly. It's not like a fraternity vibe or something, college vibe.
Right. Yeah.
Okay.
And so I think that that has pros and cons. Like I definitely miss kind of interacting with undergrads as much as I did when I was a Bauer fellow, and we had two really amazing undergrads work in the lab, and so I've tried to. UCSF has a lot of programs to bring people in from San Francisco State or from the City College, and so we're trying to still connect our lab to more younger scientists, and help kind of boost their careers.
That's awesome. Good. At UCSF, you kind of continue your research and expanded it. And I think your group publishes like 10, 20 papers, or involved in 10 to 20 papers a year or something. So it's hard to keep up, but you also helped establish two things: the No Biotic facility when you began, and then recently the Benioff Microbiome Center. Something. What's your experience been at UCSF in terms of building your lab, scaling it up? What have you learned? And then, what have you learned in terms of leading other institutions along with being a professor?
Yeah, I mean, I guess there's a lot to talk about, but I guess just in terms of like starting the lab, I mean, I kind of had a very, you know, I didn't have to kind of learn a lot of the like lab management things until I was already here, because you know, mainly grant writing was the big thing that I struggled with, you know, because you know, I mean, I did get an NSF fellowship when I was a grad student, but that's a really short application. And then I only had to write 3 pages for Harvard, and then you know, I really didn't write a lot of grants when I was at Our College. And so, you know, I think like that was maybe one of the downsides of the path I took. Like, you know, a lot of the postdocs that train in our lab now, like, you know, they've written a lot of postdoc applications, they've maybe helped with our ones, like, you know, they learn a lot of the mechanics of grant writing. And so when I started here in 2014, like, you know, I wanted to try to get support for the lab and make sure that we'd be financially stable. And that, you know, those first couple of years were pretty brutal in terms of, you know, just everyday writing grants and, you know, just constant stream of rejection. And so that was kind of the hardest, I think, part of so far of setting up the lab. I think, you know, I'd already gotten the experience through the Bauer Fellows program to like, you know, I knew how to hire people and I knew how to like, you know, get projects going and had learned something about mentoring, but yeah, the financial side was really the challenge, I think.
Oh, yeah. I think, yeah, grant writing is always and every professor you talk to, especially early on in their career, it's like, it seems to be beyond brutal, something else, something undescribable. I don't even know, I can't even empathize with you at all, but you know, you did it.
I did it, yeah, and I think the big thing that I have realized over time is like that it really is a skill.
Yeah.
You know, I think in the beginning, the thing that was very frustrating, you know, it kind of gave me flashbacks of my humanities classes in college. Like, you know, I felt like I was writing stuff and getting scores that, you know, I didn't understand what I was being judged on, you know. But, you know, it is really a skill, and like, you know, there's a right way to do it and a wrong way to do it. And so, you know, I think once I sort of figured out that there's things I can do better, that really helped, you know.
Did you have to did you have to like, did somebody tell you, give you feedback, or did you pick it up?
It was mainly just practice. Yeah. I mean, I'm a big believer in like, you know, the Malcolm Gladwell, what is it, 10 years or, you know, 10,000 hours or whatever. You know, I think you just need a lot of, you know, some people are great at it right away, but, you know, I think a lot of it is just iterating, you know, practicing writing, you know, trying things, failing, trying again, you know.
Makes a ton of sense. So maybe what's going on in the lab right now? What kind of research threads, you know, so focused on diet, metabolism, and then how it intersects with the microbiome. Anything new or anything kind of adding on top of what's been your experience from, you know, pioneering the field in like the mid 2000s to now? What do you see as the opportunities in microbiome research? What do you see as kind of the challenges and the core questions maybe to pursue?
Mm-hmm. Yeah, so, you know, our lab after we moved here kind of continued to work on food and drugs and how they interact with the gut microbiome. And then the area that we kind of started on that was really new, kind of arose out of interactions with another person here that's now a faculty member in our department named Joe Bondi Danami. So, you know, he came in and he had done a lot of research on CRISPR-Cas systems and bacteria and bacteriophage. And, you know, I'd always been really excited about phage, you know, as a way of maybe more precisely changing the microbiome. And so, you know, together, you know, we've, you know, done some initial work kind of, you know, trying to develop ways of engineering phage to target members of the microbiome and deliver genes into the gut microbiome. And then we're also starting to, you know, think more about kind of more exotic phage that you know, are normally found in the system and try to understand what determines whether or not they can or cannot act on their targets. So yeah, I think that's opened up a whole new area in the lab of, you know, thinking about viruses and you know, how bacteria defend themselves against them.
Maybe we can also talk about like there's all these connections between the human microbiome and everything from diabetes to autism. But trying to establish a causal link between populations of microbes, even certain like species of microbes, to those diseases, and like, establish a mechanism that can be drugged or modified or in some way. What's the progress there? It seems to be there's more success with like something C. diff because the link is very clearly established between the microbe and disease, right? It's in the name, versus something like obesity and diabetes where the connections are still yet to be made by groups like yours. What do you see as the kind of the big problems to surmount? Hopefully get to a place where we can know which microbes are causing which disease, and then figure out how to like modify them.
Yeah, I guess, I can sort of like tell you two things. So like, one kind of relates to like my general philosophy,
Yeah.
and then, or how I think about the microbiome, and then I can tell you kind of what we did in our lab to deal with that problem. So I guess to start, like, one of the things that kind of, I think Jeff, from the early days working in the Gordon lab, like, I think he like immediately appreciated that communities can have a function. And I've only realized recently kind of come to terms with that, because I think that a lot of us are influenced by kind of classical studies of bacterial pathogenesis, like you mentioned, C. diff, like the paradigm that I think a lot of us have in our mind is that a single microbe induces, there's like a one-to-one relationship between microbe and disease. And I think what's really transformative about the microbiome is that it's not one-to-one, that it's actually many different microbes acting together to either prevent or drive pathogenesis. And I think that really requires you to kind of rethink how you study the system. And maybe it's, there's an analogy in human genetics, right? Like, in genetics, like, I think a lot of people are now comfortable with the idea that many different genes act together to drive a disease. But that's kind of what we're trying to move, I think, moving towards as we think about the microbiome. And then in terms of like our own strategy, that's part of the reason why why I wanted to kind of focus more on pharmacology, because I think part of the issue, especially in nutrition, is that there's like so many different, that kind of diet itself is too complicated, so it's very hard to reduce it to mechanism. But if you focus on a single drug, then that really narrows the scope of what you're talking about. And a lot of drugs are only affected by a finite number of microbes. So, it allows you to kind of more quickly get to mechanism. And I think the other advantage of focusing on treatment as opposed to risk of disease is that it's a lot easier to study. You can recruit patients and study what happens before or after a treatment. Whereas it's really, really hard to study what happens in the microbiome before you ever get sick.
Interesting. I think, I can understand where you're coming from in terms of like using a drug, and then like that kind of helps you narrow down what's like causing the microbial effect. Whereas when you study something like obesity, it's amazing. Your papers are amazing, right? I can understand why they got so much like pushback in the beginning, because it's such a complex disease phenotype, and then to be able to even just narrow down to certain classes of bacteria, like pretty amazing. And
I think a general problem for, they're called complex diseases for a reason. Like, obesity is like, it's not really a single phenotype.
Yeah.
It is. It's like excess body fat, but like, there's so many different ways you can get there. And so, I think a lot of, there are diseases that are much more specific, but, for something like metabolic disease, it's really hard to kind of pull apart all the different levers that are happening.
One quick question is just like, how do you think about adding another layer of complexity, like the immune system? There's all this kind of other research around certain helper T cells in the gut playing a role in immunity and interacting with microbes. how do you wrap your head around not only how microbes interact with humans, but then how like immune cells also play a role in that whole dynamic? Because like immunity,
Yeah, I mean,
yeah,
it's kind of, I can tell you a funny story. So then, we did at UCSF, we do these pizza talks for first year graduate students where, two faculty get to tell grad students about what we work on.
yeah,
And I went first and, gave a talk, and the whole thing I was trying to get across as a concept was that the microbiome does, affects all sorts of different organs and different diseases. And so we ended up with kind of microbiome on the board and arrows to, everything you can imagine.
yeah.
And then the next speaker was Ari Malofsky, who's an immunologist here, just stood up and erased microbiome and wrote immune system. But yeah, I mean, maybe it's like part of what your training is and kind of what direction you're coming at the system from. But, I mean, I think of the immune system in a lot of ways of being controlled by microbes. And so, they're kind of, able to adjust the levels of different cells or the expression of different cytokines, or trafficking of cells, and kind of indirectly through the immune system have an effect on disease. So yeah, I don't think it's necessarily like an either or.
Interesting. Yeah, I just from my perspective, immunity is one of the hardest like things to wrap your head around. It's all these different cytokines and immune cells, and so to try to like, and then the microbiome is really complex. So just try to like when things intersect, it gets really hard. And I mean that's like your career is acting at the intersections and then like paving your own path at the point where it's really hard. But maybe
yeah, and it's hard to kind of like, I mean, I think what I find difficult about immunology is that there's so many kind of layers of jargon that it's very hard to like kind of penetrate it.
I did a class
in a way that is understandable.
And it was so hard. I didn't like IL-6R, IL-6-7. Which one's which? And some are inhibitory, some aren't, and depends on which cell. And it was just too much for me. But personally, it was way too hard.
But a lot of immunologists I talk to say the same thing about microbiology. So we have our own barriers.
But you can tie it all together, and you've had this really. I mean, you were at the right place at the right time in Washington, and that really set up your career arc. And I'm sure you have so much more left to work on, and you have all the creativity to explore new areas of the microbiome and probably more. Any kind of key lessons you've learned so far in terms of, choosing a career, what projects to work on, who to work with, things like that?
Yeah, I mean, I think like, well, it really, people are motivated by different things. So for me, like, I was excited about science because I felt like I could be creative and kind of, freely explore new areas. And so, that's still what really motivates me now. I really like, working, like you said, with like people in different disciplines, like, learning about immunology or learning about neuroscience or, learning about a new type of disease. And that, kind of pushing the microbiome into new areas. Phage biology. And so, it's fun to like, to have a job where you get to learn all the time. I think, I guess it's also, I'm fortunate that I was around people that were really encouraging and kind of, helped cheerlead me through the hard parts. And I think as a junior faculty, it's really important to, to get mentors that are going to help you through the hard times, and or even friends that are not scientists that can tell you it's going to work out. But yeah, I don't know.
That's awesome. I think you chose the right people to surround yourself with, and it seems also a common theme is practice. You just keep it, right? Just keep on doing it and practice. Because I know most people, excluding myself, if something's not working out perfectly, I tend to like try to get like a survey of people. Hey, what do you think? What, is what I'm doing? Is it am I in the right direction? Just hearing you out seemed like you just keep on. You kind of you have a good nose from knowing which direction to go into and just keep on going at it. I think that's kind of hard sometimes. Is like knowing if you go in the right direction at all. I think
right. Yeah, I definitely think talking to people helps. Although, like, I also feel like one of my I guess strengths or weaknesses, depending on who you're talking to, is that one of my friends used to or said to me once that I have a really low activation energy. I mean, it really doesn't take that much. Like, if I get a little bit interested in something, like, I'll just do it right away. I'm really a doer. I like to be busy. I like to kind of push projects forward. I'm not the type of scientist that kind of sits back and ponders. And so, I think that has been helpful in that we're just always kind of pushing the projects forward. The downside is that maybe we miss the really exciting questions because we're not planning enough. But
the point of just doing things. Do you have like a hobby on the side that's like related to that? Do you like motorbiking or something? Like do something really kind of extreme or what kind of hobbies do you have outside of the lab?
Yeah, I don't know if I'm that intense in my normal life, but we have 2 kids, and
awesome.
Then I have a podcast now, which is sort of a side hobby. Science is fun, and I also am really into movies, so I watch a lot of movies.
Cool.
But
last point here is so Peter runs one of the best podcasts I listen to, where he's the inventor, he's the professor. He actually can go deep into the science incredibly more than anybody. So we'll put in the link and we're gonna promote it. Science is fun. Peter did all these interviews during COVID, and I think just being consistent is so hard. So hard to be consistent on this because we all have day jobs, right? And so to just do this on a regular basis, but listen to Peter's podcast, Science is Fun, and we'll put it in the description. Like I listen to like 5 podcasts, and like you're one of them. So like, it's like really good.
And if anybody out there wants to be the producer,
funding. I'm gonna help you with funding. I have a friend. I have somebody to help you with funding, and then get a whole studio and an editor, and I think HHMI could do it too. I think they would be interested in that. But Peter, thanks for doing this. I know you're super busy, you have kids and all. Really appreciate you taking time.
Absolutely.